Dear friends, welcome again. Dear friends, in our today's lecture, we are going to study about inheritance of X-linked character as well as inheritance of Y-linked character. In our previous lecture, we people have studied about the X-linked inheritance and Y-linked inheritance. But now, today, by considering actual traits, by considering actual X-linked character, we have to understand the mechanism of inheritance of that X-linked traits and Y-linked traits. So, let us talk first of all about the inheritance of X-linked trait. Inheritance of X-linked traits, X-linked characters. Now we all are known. X-linked characters means the characters controlled by X-linked genes and the genes which are present on the non-homologous part of X chromosome are known as X-linked genes. Now here we have to understand mechanism of hereditary transmission, that is inheritance of X-linked characters. Already we have studied there are so many examples of X-linked characters and mostly the recessive X-linked genes are responsible to develop different disorders in mankind such as colorblindness, hemophilia, myopia, muscular dystrophy, etc. In this topic, we people have to study about the inheritance of two different X-linked characters. First, colorblindness and second, hemophilia. आजच्या आपल्या या लेक्चरमध्ये आपल्याला X-linked कॅरेक्टर्सचं तसंच Y-linked कॅरेक्टरचं पुढच्या पिढीमध्ये संक्रमण कसं होतं हे शिकायचं आहे. आता जसं मी म्हटलं की अनेक वेगवेगळे X-linked कॅरेक्टर्स आहेत. ॲक्च्युली हे X-linked कॅरेक्टर्स रिसेसिव्ह जीन मार्फत जर कंट्रोल केले जात असले तर त्या मोस्टली डिसऑर्डर्स आहेत आणि त्यांची उदाहरणं मी आता सांगितले. कलर ब्लाइंडनेस हे X-linked रिसेसिव्ह कॅरेक्टर आहे. हिमोफिलिया, मायोपिया, मस्क्युलर डायस्ट्रोफी, all these are the examples of X-linked recessive characters. Actually, these are X-linked disorders. So first of all, let us talk about color blindness. As I said just now, that color blindness is X-linked recessive disorder, X-linked recessive trait. It means the color blindness, this trait, this disorder is controlled by X-linked recessive gene. First of all, let us try to understand about this color blindness in general. Name itself indicates color blindness. It is a disorder in which person cannot distinguish few or all colors. It is a disorder in which person cannot distinguish either few or all colors, and all colors appears to him as a gray color. Color blindness हे असे डिसऑर्डर आहे की ज्याच्यामध्ये व्यक्तीला कोणतेही दोन किंवा अधिक रंग ओळखू येत नाहीत किंवा सर्वच्या सर्व रंग त्याला ओळखता येत नाहीत आणि सर्व रंग त्याला राखाडी अशा प्रकारचे दिसतात. On the basis of which the color blindness is of two types. याच्याच आधारावर कलर ब्लाइंडनेसचे दोन प्रकार पाडले जातात. such as the monochromatism. Monochromatism. It is also known as total color blindness. Name itself indicate again. Total color blindness means it is the disorder in which person cannot distinguish any color. या प्रकारच्या कलर ब्लाइंडनेस मध्ये तो व्यक्तीला कुठलाही रंग ओळखता येत नाही. Such persons are said to be total color blind. This order is known as the total color blindness. While The second type is known as dichromatism. Dichromatism is also known as partial color blindness. It means the person can distinguish few colors. There are two main types of partial color blindness, such as the red-green color blindness. It is the most common type of color blindness. Red-green color blindness is most common type of color blindness in which person cannot distinguish between red and green color and both color appears to him as gray. Red-green color blindness मध्ये तो व्यक्ती, तो सफरर रेड आणि ग्रीन हे दोन कलर्स एकमेकांपासून वेगळे ओळखू शकत नाही आणि दोन्हीही रंग त्याला ग्रे राखाडी रंगाचे दिसतात. Another type of partial color blindness is blue yellow color blindness. Means in this type that person cannot distinguish between blue and yellow. Now let me tell you, we are going to study about the structure of eye later on. But you must know that human eye has a photosensitive layer known as retina. Retina, it is the photosensitive layer of human eye. त्याच्याबाबत आपण नंतर शिकणार आहोत. But this retina consists of two main types of cells. आपल्या डोळ्याचा रेटिना या नावाचा जो पडदा आहे त्याच्यावर दोन प्रकारच्या सेल्स आढळतात, नेमली रॉड सेल्स अँड कोन सेल्स. त्यापैकी कोन सेल्स ज्या आहेत त्या कोन सेल तीन प्रकारच्या असतात अँड दॅट कोन सेल्स प्रेझेंट ऑन द रेटिना ऑफ ह्युमन बीइंग contain one pigment known as iodopsin, and that cone cells are sensitive for different colors. Means cone cells are responsible for normal color vision. याचा अर्थ रेटिना वर असणाऱ्या ज्या कोन सेल्स आहेत त्या नॉर्मल कलर विजन साठी जबाबदार असतात. मात्र sometime we know that due to the recessive gene, due to the mutant gene, the cone cells are not properly developed, or we can say the cone cells are reduced, undeveloped, and therefore that person shows partial or total color blindness. Now we have to study about the red-green color blindness. The red-green color blindness, this disorder is controlled by X-linked recessive gene, that is, suppose small c, which is responsible to develop color blindness, while the X-linked dominant gene, say capital C, is responsible for normal color vision. Okay, capital C is dominant gene for normal color vision. याचा अर्थ हा जो कॅपिटल सी डॉमिनंट जीन आहे तो रंगदृष्टितेसाठी जबाबदार आहे. अर्थात हा जो जीन त्या व्यक्तीमध्ये असेल तर त्या व्यक्तीला नॉर्मल कलर विजन असेल, आणि जर स्मॉल सी हा रिसेसिव्ह जीन असेल तर डेफिनेटली दॅट पर्सन हॅज अ चान्स ऑफ कलर ब्लाइंडनेस. नाउ वी नो दॅट these capital C or small c, these are X-linked genes, so obviously these are present on X chromosome. अर्थात हे X linked जीन्स आहेत, म्हणजेच हे जीन्स X क्रोमोझोम वर आहेत, नॉन होमोलॉगस पार्ट ऑफ X क्रोमोझोम वर आहेत. सो इन मेल वी नो as there is a single X chromosome, so in male individual these genes are also present in single dosage condition. We have already discussed, and that's why the male individual frequently show color blindness. Now the different types of individuals can be identified in a population on the basis of presence or absence of these genes. हे जीन्स कोणते आहेत, कोणामध्ये कोणते आहेत याच्या आधारावर वेगवेगळ्या प्रकारचे इंडिव्हिज्युअल्स आपल्याला आढळू शकतात. लेट अस टॉक. से हियर आय एम गोइंग टू शो इन टॅब्युलर फॉर्म. कन्सिडर द मेल अँड फिमेल. वी ऑल आर नोन the chromosomal constitution of human male is 44 plus XY, 44 autosomes, XY, sex chromosome. As we are going to talk about sex-linked gene, so simply here consider the sex chromosomes, that is X and Y. Sex chromosomes in female are XX. Now, if in male individual normal dominant gene is present on the X chromosome, then such male with genotype X capital C Y is considered as normal male, that is the male with normal color vision. ज्या मेल इंडिव्हिज्युअल मध्ये X क्रोमोझोम वर हा डॉमिनंट जीन प्रेझेंट असेल असा मेल हा नॉर्मल कलर विजन दाखवेल, आणि ज्या फिमेल मध्ये दोन्ही X क्रोमोझोम्स वर डॉमिनंट C जीन जर प्रेझेंट असतील तो ऑब्वियसली सच फिमेल इज सेड टू बी नॉर्मल फिमेल. नाउ इफ इन मेल दिस gene is in recessive condition, then such male is said to be colorblind, isn't it? Because we know that in male individuals, as there is only a single X chromosome, and therefore to suppress the effect of this recessive gene, there is no dominant allele on Y chromosome, and so such male having only single recessive gene shows color blindness. While in female, if both the genes are in recessive condition, both the genes are in recessive condition, that is small c, then such female is said to be color blind. Now, if in female one X chromosome has dominant gene and one X chromosome with recessive gene, then such female, heterozygous female, is phenotypically normal. Means such female phenotypically externally appear normal, but genotypically it is said to be carrier. अर्थात अशा फिमेल मध्ये डॉमिनंट सी जीन एक असल्यामुळे हा जो रिसेसिव्ह जीन आहे तो एक्सप्रेस होणार नाही. पर्यायाने असा फिमेल मध्ये नॉर्मल कलर व्हिजन असेल, पण फिनोटिपिकली जरी ती नॉर्मल वाटत असली तरी सुद्धा तिच्यामध्ये स्मॉल सी हा कलर ब्लाइंडनेस साठी जीन आहे, म्हणून अशा फिमेलला नॉर्मल फिमेल न म्हणता सच फिमेल इज नोन ॲज कॅरियर फिमेल. Such female with heterozygous condition is called carrier female. There is no question of carrier male. Carrier male चा प्रश्नच असू शकत नाही, because we know in male individuals there is only X chromosome. Okay. Now we have to understand the actual mechanism of transmission of this trait, that is color blindness, from one generation to the next generation. आता प्रत्यक्ष आपल्याला समजून घ्यायचंय ही कलर ब्लाइंडनेस ही ट्रेट एका पिढीमधून पुढच्या पिढीमध्ये कशा प्रकारे संक्रमित होत असते. नाउ लेट अस कन्सिडर द क्रॉस बिटवीन टू पॅरेंट्स. क्रॉस फर्स्ट. इफ कलर ब्लाइंड मेल marries with normal female, genotype of color blind male X small c Y, genotype of normal female X capital C X capital C. Now when color blind male produces male gametes, these are of two types. You know, 50% of the gametes, half of the male gametes with X small c and half of the male gametes with Y. On the other hand, as the female is normal, homozygous, and that's why all the gametes produced by such normal female are of same type, that is with X capital C. Now, after fertilization, the F1 individual produced are like this: X capital C, X small c, X capital C Y. Now here you can say, here is a female, here is a male. Now this female is having one dominant gene and one recessive gene. So such female is known as the carrier female. And the male individual has a dominant gene, therefore such male is called normal male. Normal male. It means in F1 generation that gene for color blindness get transmitted from male to the female. That is from grandfather to his daughter, or we can say male to the female. Better to say temporarily male to female. Now when such normal male marries with carrier female, that is the crossing between F1. Male produces two types of gametes, X, capital C, Y. Here the female also produces two types of gametes, half of the gametes with dominant gene and half of the gametes with recessive gene. So after fertilization, let us make a checkered board to find the F2 individuals. Here I have prepared the Punnett square, male gametes, female gametes. Let us write the male gametes at the top, X, capital C, Y, and female gametes along the left side, X, capital C, X, small c. X, capital C, X, capital C, X, capital C, Y, X, capital C, X, small c, and finally X, small c, Y. Phenotype. Look, XX stands for the female, XY stands for the male. Now here in this female, both the genes are dominant, therefore it is normal female. Normal female. Here is a male with dominant gene, so it is normal male. X, capital C, X, small c, so it is female, but one gene dominant, one recessive, means it is carrier female. And X, small cy, means it is color blind male. Okay, means in F2 individual, the half of the females are normal and half of the females are carrier. Though here this female is carrier, but phenotypically it is normal. या ठिकाणी ही फिमेल जरी आपल्याला कॅरियर जिनोटिपिकली दिसत असली पण फिनोटिपिकली एक्सटर्नली ती नॉर्मल कलर व्हिजन दाखवते. आणि जे मेल इंडिव्हिज्युअल्स आहेत त्यापैकी 50% of the male individuals are normal and 50% of the male individuals are color blind. Now just trace and try to understand the transmission of X-linked recessive gene, that is small c. Look here in this cross, the color blind male with X small cy. This small c gene get transmitted to the female, and again in next generation that small c gene, recessive gene, get transmitted to the male, that is color blind male. It means this X-linked gene, it changes the sex in every generation. It get transmitted from male to female and again from female to male, and that's why such type of mode of transmission is known as criss cross transmission. त्याला criss cross transmission असं म्हणतात किंवा criss cross inheritance असं म्हणतात किंवा झिगझॅग इनहेरिटन्स असं पण म्हणतात. या ठिकाणी आपण कलर ब्लाइंडनेस या ट्रेटचं किंवा कलर ब्लाइंडनेस ही ट्रेट नियंत्रित करणारा जो एक्स लिंक्ड रिसेसिव्ह जीन आहे स्मॉल सी, तो कसा इनहेरिट होतो हे या उदाहरणामधून समजून घेतलेलं आहे. नाउ वी हॅव टू अंडरस्टँड अगेन वन मोर एक्झाम्पल ऑफ एक्स लिंक्ड ट्रेट, that is hemophilia. But before going to the hemophilia, let me tell you briefly. The color blindness, it can be diagnosed with the help of one diagnostic test called as Ishihara's test. Ishihara's test. Ishihara test was first of all introduced and discovered by a scientist, Dr. Shinobu Ishihara, near about 100 years ago. In 1917 मध्ये या डॉक्टर शिनोबू इशिहाराने ही टेस्ट डेव्हलप केली just for the identification of color blindness. In this test, in all 28 plates are used ranging from 1 to 24. In first to 17 plates, different numbers are written in different colors, and from 18 to 24 plates, some wiggly lines are drawn in different colors, and the person has to identify, person has to read that numbers on the plates, and on the basis of which the color blindness is diagnosed. Anyway, so here we have studied about the inheritance of X-linked character, that is color blindness. Now come to the hemophilia. Hemophilia. Hemophilia, it is hereditary blood disease in which the blood fails to clot, or clotting of blood takes place very slowly. All of you are known, and we all have experienced, whenever it occurs any injury and oozing of blood starts, just within few seconds that fluid form of blood undergoes some changes. It becomes thick, viscous, and it stops the bleeding. This is nothing but the clotting of blood or coagulation of blood. Actual mechanism of blood clotting, blood coagulation, we have to study in separate chapter. Circulation. Temporarily I want to tell you, सर्वसाधारणपणे ज्यावेळेला एखादी जखम होते त्यावेळेला रक्तस्राव, ब्लीडिंग सुरू होतं, आणि रक्त बाहेर पडत असताना काही सेकंदामध्ये ते द्रव अवस्थेत असलेलं रक्त हे थिक होतं, विस्कस होतं, आणि रक्तस्राव निसर्गतः थांबतो. याला आपण ब्लड क्लॉटिंग, ब्लड कोॲग्युलेशन किंवा रक्त गोठणे असं म्हणतो. नाउ इन दिस डिसऑर्डर द ब्लड क्लॉटिंग डज नॉट टेक प्लेस, or blood clots very slowly, and that's why this disease is also known as bleeder's disease. Bleeder's disease. Of course, if there is no blood clotting, then there is a chance of death of that individual. So it is definitely a fatal disease. Now, for the process of blood clotting, so many different factors are necessary. रक्त गोठण्यासाठी असंख्य वेगवेगळे घटकांची आवश्यकता असते. आउट ऑफ विच द फॅक्टर नंबर 8 and 9 if absent leads to the hemophilia. जर त्या व्यक्तीमध्ये हे दोन क्लॉटिंग फॅक्टर्स, क्लॉटिंग फॅक्टर नंबर 8 आणि 9 हे जर नसतील तर त्या व्यक्तीमध्ये हिमोफिलिया, ब्लीडर्स डिसीज हे दिसत असतं, किंवा हा रोग, हा दोष त्या व्यक्तीमध्ये दिसत असतो. लेट मी टेल यू, दिस हिमोफिलिया, दिस ब्लीडर्स डिसीज इज ऑल्सो कॉल्ड एज द रॉयल डिसीज. इट इज ऑल्सो कॉल्ड एज द रॉयल डिसीज बिकॉज इट इज फाउंड इन सो मेनी डिफरेंट रॉयल फॅमिलीज ऑफ इंग्लंड, रशिया, जर्मनी, Spain. The England's Queen Victoria, who ruled from 1837 to near about we can say 1901, she had also this hemophilia, and she passed on that trait of hemophilia in 3 out of 9 children. अर्थात इंग्लंडची जी महाराणी होती जिचं नाव होतं क्वीन विक्टोरिया, तिच्यामध्ये हा दोष होता, आणि अर्थातच तिच्याकडून तिच्या एकूण 3 अपत्यांमध्ये, एकूण 9 अपत्यांपैकी 3 अपत्यांमध्ये हा ट्रेट पास ओव्हर झाला होता, बिकॉज इट इज हेरिडिटरी ब्लड डिसीज. नाउ कन्सिडर इफ द नॉर्मल जीन फॉर नॉर्मल ब्लड क्लॉटिंग इज कॅपिटल H. कल्पना करूया की समजा H हा डॉमिनंट जीन आहे. It is responsible for normal blood clotting, and the recessive gene, mutant gene, small h, responsible to cause hemophilia. Now we know these are X-linked genes, so obviously they are present on the non-homologous part of X chromosome. So on the basis of absence or presence of these genes in male and female individual, different types of individuals can be identified in population. Capital H dominant, small h recessive. हे जीन्स मेल आणि फिमेल्स मध्ये आहेत किंवा नाही याच्या आधारावर वेगवेगळ्या प्रकारचे इंडिव्हिज्युअल्स पॉप्युलेशन मध्ये आयडेंटिफाय केले जातात. आत्ताच आपण कलर ब्लाइंडनेस बाबत हेच बघितलं. लेट अस सी वन्स अगेन. सपोज कन्सिडर द मेल इंडिव्हिज्युअल. We know again in male sex chromosomes are XY. If on X chromosome dominant gene H is present, then such female is said to be normal. And in female, if dominant gene is in double dosage condition, that is dominant gene if present on both of the X chromosomes, then such female is said to be normal. If in male this gene is in recessive condition, then such male is said to be with hemophilia. It is hemophilic male. And if in female both the genes are recessive, then it is said to be hemophilic. In female, if one gene is dominant and one recessive, then such females are said to be carrier, and we know the carrier males are not possible. Now let us try to understand the mechanism of transmission of this trait, hemophilia. First of all, if the hemophilic male marries with normal female, very simple, normal female, genotype of hemophilic male X, small h, Y, that of normal female X, capital H, X, capital H. Gametes. Two types of gametes are produced by hemophilic male. Half of the gametes with X, small h, half of the gametes with Y. And as the female is homozygous dominant, so it produces all identical gametes, that is X, capital H. Then after fertilization, the F1 individuals produced are like this: X, capital H, X, small h, and second chance X, capital H, Y. Just like the trait of color blindness, here also we can see that female is carrier because the female has one dominant gene, one recessive gene. So phenotypically this female is normal, but genotypically it is carrier. या फिमेल मध्ये हा स्मॉल एच जीन आहे म्हणून याला कॅरियर फिमेल म्हणू. and this male is normal because in this male dominant gene H is present. Now when the crossing between these F1 individuals is carried out, यांच्यामध्ये जर क्रॉसिंग घडवून आणलं तर काय होणार आहे? Let us see. As usual, gametes X capital H, X small h, here X capital H, Y. Checkerboard. I am going to show here the male gametes at the top, X capital H, Y. Female gametes along the left side, X capital H, X small h. So the genotype of F2 individuals, X capital H, X capital H, X capital H, Y, X capital H, X small h, and X small h, Y. Again here the female, this female normal, male normal, this female carrier, and this male hemophilic. What does it mean? It means that the trait of hemophilia also get transmitted from male to the female and female to the male, just like the color blindness. Means it is also the example of criss-cross inheritance. Here we have completed the study of inheritance of X-linked characters. So let us stop here today. Thank you.